We finished up with the initial segment, and now we're going to start into the conductive segment of the axon. So the axon is what conducts the action potential, and it does that through its axolemma. So an action potential involves two processes. It involves both depolarization and repolarization. If you remember that depolarization is a gaining of a positive charge through the entry of sodium into a cell, whereas repolarization is a return to that relative negative resting membrane potential because of the exit of potassium from a cell. That action potential is going to be propagated down the axon to the synaptic knob, the synaptic terminals, whatever you call them, and you'll have voltage-gated channels opening sequentially, opening one after the other to conduct that signal, that nerve signal, nerve impulse, whatever you want to call it. So the steps to it. So the first phase is depolarization, and it has a couple of steps. At resting membrane potential, the voltage-gated channels are closed. So at your resting membrane potential, that negative 70 millivolts, the voltage-gated channels are not stimulated to be open, so they're going to stay closed. As sodium starts to enter from an adjacent region, so it's going to be the region that's closer to the cell body. So as you travel down the axon, you have one segment at a time being depolarized, and then the segment right before that, as soon as it gets depolarized, will start to be repolarized. And that will make sure that the action potential only goes in one direction. So as sodium enters from an adjacent region, it will start to activate those voltage-gated sodium channels to open. As sodium starts to enter into the axon, it causes the membrane to have an increasingly positive potential until the sodium channels close, becoming temporarily inactive until it reaches that threshold, or rather until it reaches its peak when the sodium channels close off. And that just keeps on repeating, one knob to the next. And again, it kind of passes that positive signal along. And then you get into repolarization. So depolarization slowly opens potassium channels, and potassium will diffuse out, causing a negative membrane potential. The potassium channels will stay open for a longer time so that potassium can exit the cell and make it more negative than just resting membrane potential. So essentially what happens is that as you depolarize your cell, you have your sodium channels being open, and then once it reaches that kind of maximum threshold of around positive 30, but it can be anywhere from 0 to 50 to positive 50, usually around positive 30 millivolts for a nerve. That's when the potassium channels will start to open, and then you'll start to have this depolarization. They do slowly open, or repolarization rather. They do slowly open during the depolarization phase, but because it's so slow, the sodium channels their activity more than outweighs the activity of potassium channels in that depolarization phase. So you can see that this is our sodium channels opening. We have our peak where the sodium closes, and the potassium channels open, and they don't just repolarize a cell, they actually hyperpolarize it. So they stay open a little bit longer so that the cell actually goes and dips below that resting membrane potential. Eventually those potassium channels will close. Once they kind of dip down low, those voltage-gated channels will close, and the resting membrane potential is reestablished at a negative 70. So here is just a graph of that. I definitely recommend you studying this and being comfortable with it and just all the different phases of it. You have your threshold, you have your resting membrane potential line, you have the depolarization and the repolarization phases, as well as this hyperpolarization phase. So being able to read all this, understand all this, relate it to the membrane potentials, what channels are opening, what channels are closing, when the voltage-gated channels are opening and closing, you should be comfortable with all of that. Okay, so in this conductive segment, we have those two actions of depolarization, repolarization, and then there's some smaller phases within there. So there are what we call the refractory period. The refractory period is a period of time after the start of the action potential when it is impossible or difficult to fire another action potential. And it's broken up into two phases. It's broken up into the absolute and the relative refractory period. The absolute refractory period is very soon after that depolarization, and it's when the sodium channels are open and then they become inactivated. And it ensures that the propagation goes towards the synaptic knob. So essentially it's saying that when those sodium channels so again, imagine that we have kind of a full axon right here. If I want to make sure that this is my cell body and this is my terminal end, I want to make sure that the signal goes towards the terminal end. So if sodium channels are allowed to depolarize, and then they can depolarize again, that could send the nerve signal back the other way. But by having this absolute refractory period, it makes sure that, okay, this segment right here, it's going to depolarize, and there's going to be a period of time where it can't be depolarized again. It's going to close off, so it's going to say, nope, nope, I can't change again, so that way the signal has to go in one direction. The nerve, the ion flow has to influence the segment after the more distal segment rather than the more proximal segment. So you have that absolute refractory period. It's when no stimulus can initiate another action potential. It's right when the sodium channels become inactivated. And then you go into the relative refractory period. This is just after the absolute refractory period. And in this point, you can actually re-stimulate. Another action potential is possible. The sodium channels have reset at this point, but you need the minimum strength for the stimulus is now much greater, because it's during this hyperpolarized phase where the relative refractory period is. So here is this image. So right here during our depolarization, repolarization phases in this 3 and 4, this is our absolute refractory period. This is when our sodium channels are still open, so they can't be opened anymore. And this is when they're going to be inactivated, so they haven't quite reset yet. And so it's only until they start to reset, it's only until we get below our threshold value that our sodium channels start to reset and they can actually be depolarized again. But because of how the potassium channels work, it doesn't just stop at that -70, it's going to hyperpolarize a little bit. So you still, even after this absolute refractory period, have a relative refractory period that kind of gives the nerves just like a little breathing room. So it's like, okay, I'll work if you need me to, but you have to really need me here at this point. And then, okay, once I get into 6, that's when it can easily depolarize again. Okay, so how is that signal conducted? It has two different methods of being conducted, both continuous or saltatory. So the continuous conduction happens on unmyelinated axons. It happens on just exposed axons. The charge opens voltage-gated channels that allows charge to enter, which spreads to an adjacent region and opens more channels sequentially. It's still going to go in one direction. It will still have, even without the myelin, the axon will still have the absolute and relative refractory periods, so the signal will still go in one direction. Saltatory conduction. This is going to occur on myelinated axons, so that's axons that have the myelin sheath. The action potential occurs at neurofibril nodes, those nodes of Ranvier, the little spaces in between the myelin sheaths, which is where the axon's voltage-gated channels are concentrated. So in a myelinated axon, the places that have the myelin sheath, you're not going to have as high of a concentration of sodium-potassium channels as you will within the neurofibril nodes, those segments between. After sodium enters at a node, it starts a rapid positive current down the inside of the axon's myelinated region. The current becomes weaker with distance, but still strong enough to open voltage-gated channels at the next node. The full action potential will occur at the next node, and the process repeats, the impulse seemingly jumping from node to node. So when it says that the current becomes weaker with distance, it's referring to the distance within that 1 millimeter segment of myelinated axon, not from the soma, the cell body, all the way to the terminal end. It's going to the action potential. If the action potential is strong enough to be generated, it will be propagated all the way to the end. But within the axon, inside of those 1 millimeter segments of myelination, because it has a lower concentration of voltage-gated channels in the myelinated segment, the signal slows down a little bit, but it's still strong enough to move through that whole myelin sheath, and then it gets into one of those neurofibril nodes where it has all of those voltage-gated channels that can open, depolarize fully, and it will send the signal floating on down, or pushing on down. Saltatory conduction ends up being much, much faster than continuous conduction, and it uses less ATP to maintain the resting membrane potential. So here is just the graphic example of that. You have the continuous, where it's moving down in one direction, depolarizing each little part of the axolemma, whereas in this part of it, it's still kind of having depolarization, repolarization, but you have a diffusion of the sodium here. It kind of slows down, and then it depolarizes again, and it slows down, and then it depolarizes again at each one of these nodes. Okay, there. So now we're in the transmissive segment, the last portion. So the arrival of the action potential opens voltage-gated calcium channels. Remember that calcium will diffuse into the synaptic knob after those calcium pumps have established a concentration gradient. So there's more calcium in the interstitial fluid than there is in the cytosol, the axoplasm of the synaptic knob. So calcium is going to want to rush in. Once that action potential reaches this transmissive segment, those calcium channels are going to open. Calcium binds to proteins associated with synaptic vesicles to trigger exocytosis. These are, I think your book mentions them, they're synaptotagmin, synaptobrevin. They essentially act as kind of anchors. They're different types of proteins that the vesicle will have and that the inside of the plasma membrane on the synaptic knob have. And they kind of hook onto each other. They kind of twist around and wrap around each other. And they pull the synaptic vesicle in towards the membrane. And then it starts to open up. They start to kind of pry it open so that way it can release its neurotransmitter. And that's exactly what this says. It's just vesicles fused with the membrane. The neurotransmitter is released into the cleft, the synaptic cleft. You have neurotransmitter binding to postsynaptic receptors. So the neurotransmitter is released from a presynaptic neuron and it binds to a postsynaptic receptor. Okay, get that. Remember, get those kind of straight. You have the presynaptic neuron releasing the neurotransmitter, the postsynaptic neuron having neurotransmitter receptors. Neurons can synthesize more than one type of neurotransmitter. It's not like you have just one neuron for one type of neurotransmitter, but only one type is released at a time. To release a different type of neuron, it has to have a different frequency of action potential. It can't just have the same action potential and magically know what to release. It has to have a certain frequency, a certain kind of tune to be able to release the specific neurotransmitter. Here is just an example of that, the nerve signal entering, changing the membrane potential, voltage-gated calcium channels open, that binds to the membranes, which help it diffuse to, or that binds to the vesicles, which help it diffuse to the membrane. That releases its neurotransmitter into the cleft, that can bind to receptors and open those chemical-gated receptors on the postsynaptic neuron. Okay, so just a quick summary of graded versus action potentials. Remember that graded potentials occur in the neuron's receptive region due to ion flow through chemically gated channels. They can be positive or negative changes in charge, and they're graded. So they have a larger change to a stronger stimulus or a smaller change to a weaker stimulus. And they're also local. They travel only a short distance. So right in this segment where we're seeing here, this is the transmissive segment of the presynaptic neuron. This right here is going to be your receptive segment of your postsynaptic neuron. And that's for the graded potentials. And then for action potentials, they occur on the conductive region, so the axon of the ion flow due to voltage-gated channels. They involve depolarization and repolarization, and they have an all or none threshold, or an all or none response. So once it doesn't matter the signal strength, there is not a graded potential that's going to happen here. It's going to be an all or none. So if threshold can be reached, then you will have a full response. And they propagate down the entire axon. So it can be very, very long in some cases. If considering some of your somatic neurons, it can be a very long distance to travel, not just a short local distance. Okay, so the velocity of action potential propagation. So conduction speed depends on two factors. It depends on the diameter and the myelination. A thicker fiber conducts faster than thinner ones, which may be a little kind of misleading, but don't confuse thick with dense. Thick means it refers to the diameter. So the more space you have to flow through an opening, a lumen, the less resistance there's going to be. Think like if a vessel vasoconstricts, it's harder to push any kind of fluid or solutes through it, and so you have a lower flow. Versus, like if a water hose, like if you crank a water hose, you don't get as much water coming out, but if you kind of let it go, it goes shooting out a lot faster. So thick axons offer less resistance, and myelination fibers conduct much faster than unmyelinated ones. So current flow under myelin between the nodes is very fast. That is opposite from what it's supposed to be. We remember current, it starts to slow down in the myelin and then it jumps in the nodes. The book messed it up. Okay, so nerve fiber groups. Not all nerves are going to conduct the same way. So a nerve fiber is an axon and its myelin sheath. Group A are large and myelinated, and they conduct very fast. Most of these are going to be your somatic sensory and somatic motor neurons. Whereas Group B and Group C conduct a lot slower. They're small and or unmyelinated. They're some of your visceral neurons, so some of the ones that regulate our body systems, and some of the somatic sensory neurons from the skin. And there is a benefit sometimes to things changing at a slower rate or moving at a slower rate. We don't want to have crazy if our body's trying to maintain homeostasis, and assuming that we are fairly healthy, we don't want our body on a normal basis to be going through these big changes, having really fast nerve signals like we do with our muscles. We want our organs, our visceral systems, to be moving a little bit slower and to adapt a little bit slower. And having these Group B and C neurons allow for that delayed response. The impulse frequency will vary in different neurons. So although the impulse amplitude, the change in voltage, is constant, so for an active potential you have that constant change in voltage, the firing frequency varies with stimulus strength. An example of this is bright lights causing faster firing frequency in the optic nerve rather than dim lights. So again, I always use, because I have a light right here, I have this light shining my eyes, it's nice and bright, so it's firing more frequent signals. When the clouds or when the clouds cover the sun, it's still bright out, I'm still being able to see it still lit well, but it's not as bright, and so it's not having as frequent of a stimulus. When motor nerves fire at a faster frequency, it causes the muscle to generate more tension, and so that's just another way. The frequency of firing will cause the muscle tension to change. For neurons that can use different neurotransmitters, the firing frequency can influence the type of transmitter released. So we said that a postsynaptic or a presynaptic neuron can synthesize different types of neurotransmitters. It doesn't have to synthesize and release just one. And depending on the type of frequency, that the frequency of signals that it receives, it will release a different type of neurotransmitter. Okay, so neurotransmitters, what are they and how do they work? So neurotransmitters are synthesized by neurons and then stored within vesicles in the synaptic knob. They are going to be synthesized in the soma. Remember, the soma is where everything occurs, and then you have that anterograde, retrograde transport that moves things back and forth, the fast axonal versus slow axonal, you remember. So the neurotransmitters are synthesized in the synaptic knob, and they're transported down within vesicles they're synthesized in the soma and then transported to the synaptic knob and vesicles. They're released from there when the active potential is received, bind to the target cell, and they trigger a physiologic response in a target cell. It could be a gland, it could be a muscle, it could be whatever. There are approximately 100 different neurotransmitters that have been named and classified. So the chemical classes of neurotransmitters, you have acetylcholine, biogenic amines, amino acids, and neuropeptides. We've probably all heard about acetylcholine. We talked about that when we did our muscle lectures. It has a very different structure from other transmitters, so it's on its own little class. Biogenic amines are an amine that has been slightly modified to synthesize the transmitter. This includes your catecholamines, indolamines, made from histidine or tryptophan. These are just different types of amino acids. You have just your regular amino acids that have not been modified. GABA, though, is a synthetic amino acid. It is not a naturally occurring amino acid. But there are other amino acids that will act as neurotransmitters. This is, your book mentions this kind of briefly, that it is kind of a slightly controversial thing to how can amino acids, this basic building block of our life, also work as neurotransmitters. They're so abundant through our cells. How can they function effectively as neurotransmitters? So it's an interesting way that our body manages to do that. And then neuropeptides. These are peptides. They're just within the nervous system, so we call them neuropeptides. They're chains of amino acids, 20 to 40 long, that have your endorphins and different substances. We can classify them by function, classification based on excitatory or inhibitory signals that they have. So if a neurotransmitter excites a neuron, if it's going to depolarize it and conduct if it's going to excite the neuron to do an excitatory signal, it's an EPSP. Inhibitory transmitters called IPSPs. Some transmitters can excite targets and inhibit others depending on the specific receptor present on the target cell. We'll look at acetylcholine in just a moment and how it will react differently with depending on the receptor that it has. You can classify them by their action. So direct neurotransmitters bind to receptors that are chemically gated channels. The direct transmitters, they bind to receptors that are chemically gated channels and have an immediate postsynaptic potential. So when you think about acetylcholine binding to a receptor, it's going to be binding to, for muscle, for skeletal muscle contraction. It binds to a chemical gated receptor on a postsynaptic neuron and immediately depolarizes it. Indirect transmitters bind to receptors that involve what are called G proteins and second messengers. I believe that when we did our chapter 3, chapter 4 stuff, we talked about G proteins, or we mentioned them briefly. They activate what are called these second messengers. So G proteins sit on the cell membrane. The neurotransmitter will bind that G protein, and then within the cell, the G protein activates some other type of cycle, some other type of process that can then go on and synthesize different proteins usually. So G proteins, these second messengers, have more diverse effects possible, and they could also have a much broader effect. So while direct transmitters, they cause a direct effect to occur, the indirect transmitters binding to the G proteins, they cause this cascade to happen so they can actually be amplified. Their effect is amplified more. This is just a breakdown of that, the chemical structure and the function structure. Okay, so let's look specifically at acetylcholine. Acetylcholine, it's best characterized because it has all the 4 qualities of a neurotransmitter. It's used in the peripheral nervous system to stimulate skeletal muscle and in the central nervous system to increase arousal. It's synthesized from 2 molecules, acetate and choline, that are stored in synaptic vesicles. Some acetylcholine attaches to the postsynaptic receptor. It's only brief. It does not stay bound for very long, or else you would have this kind of continuous muscle this kind of almost tetanus muscle contraction. And it's cleared from the cleft by acetylcholine esterase, that enzyme that breaks it down. They are then, it breaks it back down into acetate and choline, and that's taken back up by the presynaptic cell for recycling, where your acetylcholine is resynthesized. The effect on target cell depends on its receptor. So you hear a lot about nicotinic and muscarinic receptors in reference to acetylcholine. So a nicotinic receptor is going to have a direct binding to acetylcholine, and it will be always an excitatory postsynaptic potential. It will always induce an EPSP. So if you have acetylcholine being released from a presynaptic neuron and it's binding to a postsynaptic neuron and it binds directly and it exhibits an EPSP, then that is going to be a nicotinic receptor. If it's a muscarinic receptor, it's going to bind indirectly. It's going to act indirectly rather, and that means that it will go through that G protein cascade, and you'll have either an EPSP or an IPSP. So nicotinic receptor, direct, EPSP, muscarinic receptor, indirect. It could be EPSP or IPSP. So the removal of neurotransmitter from the synaptic cleft can occur in varied ways. The enzymes might degrade the neurotransmitter, like the acetylcholine esterase. The presynaptic neuron transporters might import neurotransmitter, so they might actually have how they have calcium pumps to pump calcium out of the synaptic knob. They can have pumps that will bring neurotransmitter back into the synaptic knob. And some neurotransmitter diffuses away from the synapse, and it's resorbed by the glia, by the cells of the nervous system. So it is released in that synaptic cleft, and it just kind of floats around in there. Most of it will go and bind to the receptors, but then some of it will float away. Some drugs have their effect by influencing transmitter removal. So SSRIs will inhibit the uptake of serotonin from the synaptic cleft to help decrease depression. Galamate hydrobromide is an acetylcholine esterase inhibitor used to treat Alzheimer's disease. MAOIs also will help to treat with pain. We take those. You see MAOIs all the time. So here is just kind of that essential what we were just talking about, how acetylcholine can bind directly or indirectly, and how it has these second messenger signals when it binds indirect versus a EPSP always when it binds direct. Okay, neuromodulators. So neuromodulators are chemicals that alter responses of local neurons. They can either facilitate or inhibit local neurons. Facilitation essentially means that modulation causes a greater response in postsynaptic neurons and may increase the amount of neurotransmitter and cleft or number of postsynaptic receptors. So facilitation essentially is just saying that it's facilitating the action. It's facilitating whatever the signal is going to be to make it easier for that signal to be propagated along. You can think about facilitating a muscle. So if let's say that I wanted to pick something up before I go to pick up a big heavy box, I'm going to go ahead. My muscles will go ahead and be primed. They're going to know, okay, this is going to be heavy. I should go ahead and be a little bit kind of toned and have some tension, so that way it's easier to pick this thing up. We talked about how muscle tone in our body, just our regular resting muscle tone, allows our muscles to be facilitated. That way we can have our quick reflexes. Because if there wasn't already a little bit of tone, a little bit of facilitation to our muscles, they would fire much more slowly. So by having facilitated neurons, it just ensures that signal is propagated even faster. The opposite can happen. You can have inhibited neurons that will make it harder for a signal to be propagated. They work in the exact opposite way of the facilitation. They cause a weaker response. They may decrease the amount of neurotransmitter in the cleft or the number of postsynaptic receptors. They may increase the amount of enzymes breaking down neurotransmitter. Who knows? A couple of different ones. Nitrous oxide, it could be a neurotransmitter or a neuromodulator. It's a short-lived nonpolar gas. It's made and released by postsynaptic neurons in the brain, where it's believed to strengthen memory. Its effects in the PNS include blood vessel dilation. If you go to the dentist, they'll give you nitrous oxide if you're very stressed. Endocannabinoids have influence the same receptors that marijuana does, the THC from marijuana does. No surprise, they influence memory and appetite. Okay, last bit is neuronal pools. So our body is not made of just single neurons that are running one after the other in these nice straight lines. There are different configurations for neurons. So the different circuits, that we call circuits, they're specific patterns of arrangement. You have converging, diverging, reverberating, and parallel after discharge. So probably converging and diverging, you can go ahead and picture what those are going to look like, but the reverberating and the parallel after discharge might be a little different. So for the converging circuit, exactly what you think. You have a single input converges at a single postsynaptic neuron. So you have all these different sources of input and then one source of output. The example that it gives, multisensory input, synapsal neurons in the salivary nucleus. Sight, sounds, and smells of cooking lead to one output, salivation. So you have all of this different type of sensory input coming into one effective output signal. A diverging circuit, you will have, it's spreading information. So you have one presynaptic neuron to several postsynaptic neurons. This is similar to your brain kind of controlling and commanding yourself as you walk. So you have this kind of one kind of commanding neuron that's going to be sending out a lot of different signals to your leg muscles, to your postural muscles, to your muscles in your eyes, helping you to look, in your head to make sure your head is balancing correctly. All of those different things that have to happen when we walk that we really don't think about, but really do take a lot of different coordination. The reverberating circuit is a feedback to produce repeated cyclical activity. It's helpful in breathing. It's one of those things where you don't really think about breathing, but breathing is really important in maintaining blood volume or proper blood chemistry. And so it's this feedback circuit that's just kind of constantly going that we don't have to think about, but it just has this different inputs, different outputs to help maintain that proper rate and depth of breathing. Once these start, they stay active until there's an inhibitory stimulus or synaptic fatigue. So inhibitory stimulus meaning that something finally says, nope, stop, don't do that anymore, which could be you kind of just, it's one of those things where you don't really realize you're breathing, and then as soon as you think about it, you take a big breath and kind of get back into it. Or synaptic fatigue, meaning that they just run out. They run out of neurotransmitter, the receptors start to become overloaded and they can't bind anymore a neurotransmitter, and the circuit can no longer continue. The last one is the parallel after discharge. This is input being transmitted simultaneously along several paths to a postsynaptic cell. So you have different sources of starting input that are also communicating with each other at different points, coming into this one output cell. The signal arrives at various times, and so I like the book's description of an echo. You kind of just get these little signals and sounds that say, okay, here's what we need, here's what we need, here's what we need, at different times though, and so that kind of gives your body or your nerves a greater sense of, okay, well, what's instead of just here's this one moment in time, here's several moments in time, let's process all of that. This is obviously then involved in higher order thinking. This is going to be a much more complex type of circuit, and so it's involved in higher processes. And that is all of chapter 12.